Thanks for joining me in this presentation. So, I urge you to have a quick look through the legal disclaimers. They are a little bit important, but over to the kind of main, if you like, presentation. It's a little bit about us. We're an Oxford-based, AIM-listed computational biology company, recently added an RNAi drug modality platform, which I'll be going through, and also going through some of the commercial collaborations we have with kind of top-tier pharma companies like Novo Nordisk, MSD, and Galapagos. And, the reasons why they would want to sort of collaborate with us. So, the kind of slide says, what do we do? We construct the most accurate in silico, or if you like, computational models of any biological function. Actually, we can model even a sort of simple biological function such as eye color, not that simple, but obviously we're much more interested in disease state biological processes and functions in order to be able to kind of analyze them, etc. So you can think about e-Therapeutics as having a sort of computational or in silico laboratory of biological functions. So once these kind of models are established and they're accurate, we go on to be able to then use them to identify novel drug targets and biological mechanisms. We then go on to design intervention strategies, such as using novel small molecules and the recent announcement of the RNA platform. Okay, so on the left-hand side, you can see some examples of what these kind of models look like. This is a model we did in SARS-CoV-2 hyperinflammation. This is one of insulin resistance and type 2 diabetes. As you can see, a real kind of diverse biological areas. And the models, if you like, what are they really? They're the nodes you can think of as genes or proteins. Now, going back to my example of a very simple thing such as eye color, in fact, it's really not that simple. Eye color in of itself is not controlled by one or two genes or proteins. That would make kind of eye color quite susceptible to damage. In fact, maybe something like eye color or any of these kind of biological functions are networks of proteins or genes which are kind of collaborating with each other and forming this kind of unit, which is, one of its main functions is to be, incredibly sturdy and resistant to biological attack. So these are our models. How do we design these models? Where do they come from? We use our computational platform. So as a starting point, we have the most complete human and animal protein-to-protein interaction database where, we have kind of every recorded function and communication between proteins in humans and animals. Like all these types of data sets, some aspects of the data are excellent, or complete, or very good, but there are going to be inevitable kind of missing data sets. That's where our machine learning algorithms kick in to basically make predictions for where there aren't incomplete data sets. So on top of that, we use techniques such as what's called natural language processing systems, which are in effect ways of extracting large amounts of data. We have access and are able to, if you like, download, analyze, and curate every paper that's been written in almost any disease area or biology, any experiments that have been done, and we're able to do that and kind of like store them and analyze them in a very fast and efficient manner. And I guess the third kind of component of our computational platform is we have the most complete database of small molecule compounds, something in the region of 16 or 17 million of them. So, if you think there are something like greater than 3,500 approved small molecule compounds, and they'll have the very best annotations attached to them. They're approved drugs, their mechanisms and activities are well known. And then the rest of the several million compounds in there, again, range from very good annotations, very good information attached to them into their activity, all the way to very poor. And in the zones of very poor, or in the middle sections, is where our machine learning algorithms again start to predict their activity. So think of our sort of computational platform as a kind of a series of databases enhanced by machine learning where there are gaps, and an ability to be able to capture data using techniques such as natural language processing. So some of the outputs from the platform, what do you get out of it? As I said earlier, targets, compounds, and genetic insights. That's just some of the things that you can do. As an example of targets, so we did some work in SARS-CoV-2. We generated network models of all the genes that we think are causal in this, kind of creating this mesh that you can see with the different connectivity. We then sub-analyze these to find the very key proteins or key functions. What are the key drivers of SARS-CoV-2? And from there, we can generate novel targets to be able to kind of attack the SARS-CoV-2 virus. An important part of our offering is compounds. So that's using our small molecule compound database. So, again, I've given an example here of insulin resistance. This is a sort of template network of insulin resistance. So you can think of what we do here as almost like a little bit of a computer game. We use our compound library to attack the network. The idea being the compounds which are most potent will disintegrate these networks. We will then list the best compounds, let's say the top 300 in terms of which ones are the most active. We'll come through some of the performance metrics in the next slide. We take these compounds, we find what targets they're working on. This is all in silico. In terms of genetic insights, this is particularly interesting for the outside world at the moment and for our RNA platform. So, population-wide genetic studies, tens of thousands of people, let's say in Alzheimer's, the outputs of those are usually a list of, let's say, 200 or 300 genes, which are shown to have some cause in, let's say, Alzheimer's. But obviously 200 or 300 genes are not really actionable. There's just too many of them. And in fact, these genes individually have a very small part to play. Again, we kick in with our network models where we can connect these genes up in terms of their communications. From the networks, we again look at the sort of key processes within those networks, understand what novel targets we can actually go and attack with drug modalities. And an important part, which I've tried to sort of depict with these graphs here, is you can think of the kind of computational or in silico function as something which can de-risk, find the best biology, best mechanisms, the best targets. But eventually what we will do is take all our outputs into what we call wet lab settings and test them in real biological models. So our platform has had many kind of internal and external validations. On the left-hand side here, you can see the different projects we've done. We've generated really millions of different networks of biological functions to various degrees. Here are some that we've developed even deeper than those. We've gone potentially all the way to kind of preclinical studies of them. So we've gone from, if you like, ground zero in the computer all the way to animal studies and even potentially commencing phase 1 studies. And you can see the diversity of disease area where this axonal degeneration is obviously a central nervous system potential mechanism, influenza, which actually kickstarted our SARS-CoV-2 work. And you can see in our partnerships, again, very diverse disease area, type 2 diabetes, CNS, and fibrosis with the likes of Galapagos. So in terms of performance, if you like, one of the good outputs and key outputs that we sort of really talk about is on this section here. Just take a step back, a traditional pharmaceutical process would be more akin to, a big pharma company will set up an assay which is trying to basically mimic, let's say, insulin resistance. Then they will use robotics technology where they will, up to potentially even 500,000 compounds, where they just test these compounds in a blind fashion to find out which compounds have activity in their assay. Anything that shows activity is called a hit. Over here, you can see that even with a very, very large number of compounds, 500,000 or 1 million, the hit rates can be 0. You can find no hits, and that's obviously an expensive exercise. And a small percentage will generate hits, i.e. they'll show some activity, but they then go on to the next problem is they found the compound, but it's biologically blind. They now have to go all the way back, find out what target it's actually acting on, what the sort of biological relevance is. So that's only the kind of starting process. Whereas if you go through our kind of, if you like, in silico laboratory, you are coming at it, you're looking at your biology in a kind of rational sense, you're building network models of them, you understand mechanisms and targets first. You then, using our compound database, generate a very, very small number of compounds, something like 1,000 are generated. So you only have to test 1,000, sometimes even 500. And our hit rates, across the board have been, 100 to 1,000 times order of magnitude to industry hit rates. So the ability to be able to model biology and to be able to intervene in a kind of very, very precise and much more sort of rational sense is a big kind of unique selling point of our platform. So external collaboration is very important to us and a big part of our business model. Novo Nordisk, MSD, Galapagos. We're talking and having ongoing business development discussions with other partners, looking really to do bigger transactions where, what I kind of would call enterprise deals, where we can sort of really get into perhaps giving away one very big disease area, but working with, the expert pharmaceutical companies in that area. The economics are better, but also our ability to stress test our systems and to be able to improve them will be kind of exponentially beneficial to us. Okay, so I'm going to spend a few slides on our new RNA platform announced a couple of weeks ago. Kind of precursor, why did we do this? I guess headline sort of things would be, well, kind of first and foremost, RNA drugs are amongst the most sought after drug platforms across the pharmaceutical industry today. So for example, as you know, the current main vaccines are RNA-based vaccines, and I'll come on to that in a minute. The second aspect of it is we've got a kind of very, very lucky or unique combination of people who've been there and done it in the RNA field and have gone through this process many times, and that know-how and knowledge has really turned something which could take years to produce in really a matter of months. And third of all, we thought and are very confident that the really kind of end differentiator will be our computational approach, which will really enhance our kind of offering to potential partners. We're not just coming with a toolkit in terms of being able to do things with it, but we also come with information about the targets and mechanisms that I discussed. So since, as I said, RNA kind of therapies seem to be extremely popular, obviously mainly because of the kind of mRNA vaccines that people are taking every day, it seems now. And I thought I'd just be a good contrast if I could just give a contrast between difference between an mRNA vaccine and, if you like, our RNA platform. So kind of in simple terms, all RNA drugs have two key components to them. The drug is a genetic sequence. You can see an example of our genetic sequence here. And the second component of it is once it's successfully introduced to a cell, the genetic sequence is actually processed by the cell itself. So the drug-like effect is actually created, if you like, in your cell by the cell itself. In the case of the mRNA, to just give you a very quick walkthrough of what happened, and part of the reason it's happened so quickly is some of you may know SARS-CoV-2 uses what's called a spike protein to infiltrate our cells. You can think of the spike protein as its kind of landing gear, if you like. So the first step for the likes of Pfizer, BioNTech, and Moderna was to find the genetic sequence of the spike protein, which was very easy to do. The next component was to find what's called a delivery system to be able to take this genetic sequence into our cells. And in the case of the vaccines, it's something called a lipid nanoparticle. Once that is created and once you get the injection, the delivery system will put the genetic sequence in your cell. And as I said, the minute the genetic sequence is in the cell, your cell's machinery will read that gene and make the spike protein. When the spike protein is made, your immune system kicks in, spots a new entity, if you like, and starts to generate immunity. Our process, RNA platform, very, very similar, but in some ways the flip side of it. Firstly, this weird-looking cartoon here is our delivery system, which takes our genetic sequence purely and only to the liver, and I'll come to that in a moment. And our process starts with, we look for a target gene in the liver which is causing a disease. We then go and find that genetic sequence of that target gene, which is very easy to do. We make our genetic sequence. Upon injection, this drug-like element, if you like, it goes into a cell. Once it's into a cell, the cell itself processes it, finds the gene that we're interested in, and shuts down that gene's activity. So if you want the kind of difference between an mRNA platform and what's called an sRNA platform, is that an mRNA vaccine, mRNA drugs make proteins, we shut down proteins. Okay, so what are some of the other filters to get into this space? RNAi in the liver has kind of some very unique drug-like properties. So the first thing I would say is got this kind of specificity upon specificity. The delivery system will only take the genetic sequence to the liver or hepatocytes. All other cell types are left uncontaminated. So the big advantage there is you target the gene you're after only in the cell you're after, and the same gene is allowed to operate in other cells untouched. You know, what you don't want to do is switch genes off in every single cell type. You want to be selective to the liver. The very, very big commercial advantage, which makes this RNA platform almost what's called gene therapy, like you get the best of both worlds. You get something which is not permanent, but what if you like semi-permanent. So with one injection, you get a very long duration of action. Actually quite nicely depicted in this chart here, but if we start here at day 1 of injection, and if we follow the purple line, this is a target gene, and you can see that its activity is being shut down around day 14. It's in effect shut down and not causing disease. But what is interesting is how that purple line for up to like 180 days, the effect is just continuous. You don't need more injections. You don't need to take a daily pill. So with one injection, you know, a patient can control a disease for 180 days or 3 months. And that obviously opens up a huge, vast swathe of diseases such as cholesterol and things like that. So obviously a very, very big multi-billion drug platform opportunity. Kind of an important, very important tick box is this is no longer in research or in terms of risk. Is now got, this area has got 4 approved drugs. And in terms of risk profile, it is the exact moment when a company like us should be entering this fray, when multi-billion dollars have been spent, you know, validating the technology. So, you know, the biggest factor for us was the significant financial deals being done in the sector. So the first thing I would highlight is, you know, the very big and trophy acquisition in the sector was Novartis paying $9.7 billion for a medicines company. And that was for one RNA drug, not even a platform. And that kind of changed the industry from, you know, something exciting to something very meaningful in 2019. But to give you an idea of the type of very early deals that, you know, are achievable, I would point maybe to this one. One of our competitors, Dicerna. So Novo Nordisk went to them and said, we want to reserve 30 genes in the liver. They wouldn't disclose which genes. And for that, they paid Dicerna $175 million in cash up front, plus any back-end royalties if Novo Nordisk was successful. You know, that's a very, very substantial deal, given that there are something like 10,000 potential genes in the liver. So for Dicerna to give 30 out of 10,000 and have payments, back-end payments, it really gives you a flavor of how early and quickly you can do deals in this area. And there are many, many examples of very similar quantum-sized deals. So a little bit about our platform. Here's just some schematics of different permutations and combinations of IP and chemistry that we have, not particularly useful in of themselves, but again, the deep know-how in the company, you know, we're testing multiple different if you like, constructs and combinations and permutations, which we're kind of very confident of working, you know, and we have a strong IP position. We've just filed a patent on the delivery system, which is proprietary to us. You know, I think at this point it's important to just give you an example of why maybe some of the biotechnology stocks, the genomic stocks, why the performance has been so exceptional. I think, first of all, the mRNA vaccines have shown the way, but this is a very good example. If I have a target gene I'm interested in the liver, I can literally here on my computer go and look up the sequence. I can make my drug on my computer point and click within a couple of days and all the moving parts that need to go with it. I can press send to my CRO or my lab, who will make that drug in 3 months and for a few $100,000, maybe even a backup one. And that drug is the same drug that will be used all the way to approval. And to put that in contrast with something like small molecules, a small molecule takes 3 to 4 years for that lead compound. So we're talking about a kind of quantum step difference in terms of timelines, costs, specificity, and the ability to be able to, at least in certain parts, completely change the timelines of drug discovery and drug development using genetic sequences, really. Next thing was the liver. Is the liver, the specificity is nice, but you could also think about it as a limitation. But actually, 72% of some statistic like that of all human genes are expressed in the liver. Only 1% of them are being addressed now. So actually, the liver is an extremely active organ for drug discovery and the type of work that we do. Importantly, I've done a chart here of all our kind of competitors, and which pretty typical in the drug industry is a lot of people going after the same drug targets. Way too many Me Too's and kind of people with very little advantage over each other, and that's again, given us and given me the kind of confidence to say, look, we've got a very, very big white space of biology to aim at here. So I think what really sort of completes the picture for me was, like I said, coming at the toolkit, which could have taken years and months, is pretty good. But I think what really gives us a different seat at the table is the ability to be able to come with an angle to the biology. So we have for a while now kind of created what we call a sort of liver-focused informatics team. And you can think of that as a sort of extract from our general platform. So we've extracted the best data in hepatocytes and the liver and created a sort of subunit of it. And here is a picture of a hepatocyte. I think even now, we now got the most complete network map of hepatocyte biology anywhere in the world. We've got the leading experts in the world in hepatocyte biology. All our kind of machine learning algorithms are being pointed in that direction to be able to really capture that specificity. Some of the plans are really pretty ambitious. One plan that we're working on at the moment is, let's say there are 10,000 genes operating in hepatocytes. We have plans to be able to, if you like, stop the activity of each and every single one, and then to see what the knock-on effect in a cell is of protein expression and gene expression of all the other proteins and genes, and really build an atlas of hepatocyte proteomics and network biology. That's it. We're going to continue our computational BD general discussions, commence our RNAi discussions, etc., etc.
Lovely, Ali, thank you very much. We are somewhat out of time, but we did have a couple of questions sent in before the webinar tonight. So I just want to get through a couple of them, if I may. The first one: small private investors were excluded from a recent offer of new shares. Will the company plan to include an open offer for the next or for any future fundraise?
I don't think that's true. We had a primary bid offering, quite a substantial one actually. So no, other than that.
Well, that's fair. So in which case, if the questioner is on tonight, you don't know primary bid, send us a message and we'll point you to the platform. And
the retail investors are very important actually in the stock market, let alone e-Therapeutics. It'd be unheard of for anyone to be excluded.
Question in tonight: how come you were able to add the RNAi platform so fast when firms such as Arrowhead have been doing this for 20 years? And where did the platform come from, and who has the IP?
Look, we have the IP. We have the. So I think what's happening. The answer to the question is the RNA field is very much about know-how and very much about people who've worked in the industry for a long time. A lot of things that have happened in the last 3 years have changed the patent landscape. I think what the big area, which is the big unmet need, is the biology.
And one more here, if I may. If bardaloxin methyl, if I pronounce that correctly, works in COVID, how do you get paid?
So this is where we did a piece of computational work, what's really called a drug repurposing exercise. Instead of using our small molecule database for novel chemistry, we looked at all approved drugs. We did a computational approach, looked at all approved drugs, and we found a drug called bardoxolone methyl, which is owned by a company called Reata. And actually, we did it for two reasons. In the height of the kind of SARS-CoV-2, fear and madness, we thought we could make a major contribution. I think, and the second part is as a proof of concept of the platform. It's a very, very neat case study of how kind of we can go from a biological function like SARS-CoV-2 and find whether it's novel chemical matter or even what's out there already. So it wasn't so much a commercial enterprise, but I haven't given up on a commercial enterprise with it either. But it's a little bit complicated. But I think at the very, very least, the case study we got out of it is very significant.
Ali, lovely. We are, I'm afraid, out of time. Thank you very much indeed for presenting this evening and giving us the update. I really appreciate that.